of it yesterday. Do I really need it? I can talk loud. I hate it. It's just gonna get taken. Can you hear me at the back?
Introduction, and it's a great pleasure to be here today at Waterloo University for Brain Day. And today I'm going to talk, because this is theoretical neuroscience Brain Day, I'm going to talk about more computational aspects of my work. You want to reshare my screen? Okay, sorry. Almost. Okay, is it okay now with the Zoom? Okay. So what I'm going to do today is tell you about some recent work that has been going on in my lab, and especially two students who recently graduated from my lab, Dr. Eshed Margalit on the left and Dr. Don Fincy. But before I get started and tell you about the current work, I wanted to share a little bit about how I got into the field in the first place. So I was in undergraduates. My parents really wanted me to do something practical. And since I was pretty good at math, they told me to study electrical engineering and computer science, which was the future, and they were right about that. But I became really bored. And the reason that personal thing, I got bored is that I found out that engineers were really into making things work and less about understanding why things worked. And I was really interested in understanding why things worked. See, so my mom also worked in the library, and she used to bring a bunch of like layperson scientific papers. And one day she brought an issue from Scientific American on the mind and the brain. And I was really intrigued by one of the papers in that issue, a paper by Samir Zeki, and I'm showing you some pictures from that paper in 1992. And basically what Samir wrote in this paper is that when you look in the world, in this rich visual input, maybe you're staring at a picture in the museum, there's a lot of intricate processing happening in the brain. So basically this rich visual input is broken down into simple components like maybe color or motion or form, and then they're processed kind of in parallel in multiple processing stages. So for example, if you think about color and motion, they're processed in V1 in blobs and interblob regions, in thin and thick stripes, and eventually in downstream regions that might be dedicated to color, maybe in V4, and to motion in V5. And what really struck me is that this was a really interesting question. So the question I'm going to address today is, why is a visual system organized both spatially and functionally? The second thing that struck me is that I have to figure out how I can address that exact question, and because this has really seemed like a topic that I wanted to spend time doing. So especially I was intrigued because these were components that I could analyze with kind of an engineering approach, like orientation or spatial wavelengths or spatial frequency. So I learned from a friend of a friend that in the Weizmann Institute of Science in Israel, there are computer vision scientists that were working with a neurobiologist, and they actually study the visual system. So I applied to graduate school in the CS department in the Weizmann Institute of Science with the hope that it would open for me a path into visual neuroscience, and it did. So when I went to grad school, I started reading real scientific papers published in real scientific journals, and this is kind of a paper that was around that time when I was doing my grad schools in the end of the 90s. This is a very influential paper by David Van Essen and Jack Kalanit, and basically they're trying to kind of use an engineering approach to explain the processing in the visual system. Note that this is kind of like a schematic. It's not really any kind of code that you could execute, but they had this very powerful idea that was also echoed in a bunch of other papers, that there might be, the vision is a very complicated task, so it needs to be processed in a series of regions, maybe forming a hierarchical processing stream, and that the visual system has evolved to have multiple processing streams that might be focused to do different behavioral tasks that can be done in parallel. For maybe there's one stream, it's called the dorsal stream, that I'm going to talk about it, that's involved in processing where objects are in the scene. And there's another stream called the ventral stream that might be involved in processing what the objects are that you're looking at. Of course, a lot has changed since the 90s. One of the things that was the advent of fMRI, we learned that there are much more than 5 visual areas in your brain. And in fact, there are more than 36 visual areas that spans occipital, parietal, and temporal lobes. And they're organized in 3 processing streams I'm going to talk about today: the dorsal stream, the lateral stream, and the ventral stream. And second of all, we have new computational tools in the form of deep neural networks that could start actually implementing these kind of theoretical ideas from the 90s, and we can use them to ask why questions about the brain, and which is what I'm going to do today. So the roadmap for today is to use this deep neural network approach to understand why the visual system is organized the way it is. And I'm going to talk about 2 case studies. One is a case study of the ventral visual stream, a pathway involved in visual recognition, and one addressing the question of why might we have processing streams in the first place. So I'm going to start with an overview of the ventral visual system, and then I'm going to dive into the more computational work. So this is an inflated view of the brain. It's an actual brain of one of my students, shown from the underside, and the gyri are in light gray, and the sulci are in dark gray. So when you look at something in the world, light gets reflected from that object. An image is formed on your retina, and then it gets transmitted through the optical tract to the occipital lobe, to the first visual area that I'm going to talk a lot about today. It's called V1, or primary visual cortex. And then there are multiple retinotopic visual areas that form a processing stream. And eventually, information gets into this yellow zone. This yellow zone is called ventral temporal cortex. It's a cortical expand about maybe 4 centimeters wide and 5 centimeters long. And in this cortical expanse, percepts might emerge. For example, you might perceive that you're looking at the bird. And the reason I'm going to focus on the ventral stream is this is probably one of the pathways that has been most studied, both in humans and in primates. And I'm going to talk about the first stage, the V1, and the end stage, the ventral temporal cortex, or VTC, today. And one of the things that we know from a large research in neuroscience is that neurons in V1 and in VTC have special properties, and that they are organized in an orderly way across the cortical sheet. So, for example, from the work of Hubel and Wiesel and later on, people have found that if you record from neurons in V1, they are tuned to specific simple features in the visual stimulus. For example, the orientation of edges, if the information is coming from the right or left eye, the spatial frequency in the image, and also the color content. And neurons with these properties are not randomly organized across the cortical sheet. In fact, if you take maybe a few millimeters of the V1 in a macaque monkey and use optical imaging, you could find that the orientation-tuned neurons form kind of smooth maps of orientation across the V1 cortical sheet. If you look at another dimension of tuning, let's say spatial frequency, you also see tuning of spatial frequency that's organized in an orderly way across the cortical sheet of V1. So basically, this is a functional and spatial organization in V1. When we think about high-level visual areas, for example, in VTC, the neurons have different features. Instead of tuning to simple visual features, they're tuned to more high-level information. And a dimension that is particularly salient in ventral temporal cortex is the information about the category of the stimulus that you're looking at. And this is evident in two kinds of ways. If you look at the distributed map of responses of neurons across ventral temporal cortex, this is an example, left hemisphere of a subject, you can see that there are different spatial maps that are consistent across items of that category. So if you show different faces, they'll get a very similar fingerprint. But if you show different stimuli, like cars, you can see that you get a different distributed pattern across visual cortex. And in fact, you can look at these patterns and decode from the pattern what is the subject looking at. And people have done this with a high level of success of over 90%. In addition to this distributed information, there are also clustered regions in ventral temporal cortex that seem to be specialized for processing information related to a handful of ecologically relevant categories. For example, in the collateral sulcus, there's a region that seems to be involved in processing places. In the fusiform gyrus, there are two patches that are involved in processing faces. In the occipital temporal sulcus, you'll find two clusters that are involved in processing words. And in addition, there's a region that's involved in processing body parts and bodies. And these are in very consistent locations across subjects. For example, I can identify a small sulcus called the medial fusiform sulcus, and I can know where your face selective region exists. So this is a spatial and functional organization of ventral temporal cortex. And one of the things that happened in the last 10 years, starting from a seminal paper published by Dan Yamans and Jim DiCarlo a decade ago, has used deep neural networks to show what kind of networks explain the brain. And in the vision, we're using convolutional neural networks. And this paper from 2014, what they did was really nice, that they trained a lot of models. So each point here is a different deep neural network model. And basically what varied about the models is how good they could actually categorize the stimulus. So this is the x-axis. And then they had some electrophysiology data from DiCarlo's lab, and they measured how well their models could explain the responses of neurons in IT. So you can see this is a model, and this is the response. The black is a response of neurons to different stimuli. And basically, the main insight from the paper is that the better the model did at object categorization, which is a hypothesized function of the ventral stream, the better it explained brain responses in the macaque infratemporal cortex. So this was really exciting, and it revolutionized the field. And basically, you can see that in this kind of plethora of many recent papers that have used artificial neural networks, and especially in vision convolutional neural networks, to understand how the visual system might work. So while these networks have been very exciting and are good at explaining function, there's a real glaring gap in what they're able to explain. They cannot explain the spatial topography of the responses across the cortical sheet because that has no effect on the model's performance. But since my interest is understanding why the brain is organized the way it is, I would like a model that explains both the function and the spatial topography of responses across the cortical sheet. Because my working hypothesis is that this spatial organization might serve a computational role. So if we actually understand what forms this, maybe we can get insights into brain function. So I'm going to introduce today a new kind of networks that we have invented. It's called the topographic deep artificial neural network, and I'm going to refer to it with the acronym TDAN. And basically, here's a schematic of our TDAN, and I'll explain to you a little bit more about it. So the TDAN is based on a convolutional neural network called ResNet. We use ResNet 18 because it has about similar layers or depths as a ventral stream. And there are two main differences about this network than the standard ResNet convolutional network, is that each convolutional layer also is associated with a simulated cortical sheet, meaning that units in this layer are actually placed on a two-dimensional grid that has physical units. And basically, these units before training are initialized with a retinotopic location. So if you see this purple patch in the image is translated to these purple patches across the layers of the network. So this is one difference, that we could actually simulate the placement of neurons or units. And the second thing is that when we're training the network, usually networks with using backpropagation have a loss function that's related to a task, maybe like a categorization task, like in the original Yaman's paper. I'm going to show you different tasks today, but what's really different about our network is that in addition to a task function, we also have a spatial loss function. And basically, what the network tries to do during training is to place model units that respond similarly on the training images, physically proximal on the simulated cortical sheet. And the intuition of this comes from this research called self-organizing maps. And the idea is that if you place neurons that have correlated responses close to each other on the cortical surface, you might reduce the wiring lengths of the system. So it serves maybe efficiency purpose. And we can govern the amount of the spatial weight with this a parameter called alpha, in which it basically tells the relative contribution of the spatial loss function compared to the task loss function. So I'm going to start today with telling you about one of the TDANs that we implemented. And I'm going to use this TDAN to see how well it explains the functional and spatial properties of V1 and VTC-like layer of the networks. And we find this by using ridge regression to find which layer in the network best corresponds to the functional properties of V1 and VTC, respectively. We trained the networks with a large image data set called ImageNet that has millions of images. And I'm going to talk about this network that works really well. It's trained in a self-supervised way. Basically, we let the network train with a contrastive task. And basically what the self-supervised task does is that it is trained with similar version of the same image, for example, shifted version of the same car. And its goal is to output a representation that's similar across different versions of this image, but very different from all other images. So this is what this algorithm does. And we used it because other people have shown that it's also pretty good at explaining the brain. And when we talk about a spatial loss function that we're using, we're going to use a spatial loss function that's correlational, and it's tied to the relative distance within an area. So it kind of scales with the size of the cortical sheet. And I'm going to show you, again, results with an intermediate spatial weight, because this intermediate spatial weight works well at explaining brain responses. Does this make sense? Any questions so far? Yeah. Yeah, sorry. So the spatial loss means R is the responses and D are the distances. So basically it's trying to put neurons with similar responses closer on the cortical surfaces. And this is a correlational distance, basically, rather than absolute distances that might have been, let's say, a Euclidean distance. Yeah.
And then, okay, so then the task is, can you repeat what the task is?
Yeah, so the SimClear is, it's called a contrastive learning task. And basically what it does is that when you train, you generate multiple versions of the same image. You can crop it, to shift it, and maybe blur it. And basically the network is supposed to give the same response to the different versions of that particular image. And this, it's called contrastive because it's supposed to be similar for that image, but different from all other images. For example, if you had a shoe or something else.
So similar in the latent space.
Yeah.
But the other one, the spatial, is more to do with this spatial organization.
Yeah.
Of the neurons.
Yeah, so this is a functional task on the visual images, and this is really trying to get model units that have correlated responses on the training images to be physically proximal on these simulated cortical sheets. Basically, you want to put them nearby on the simulated cortical sheet. Yeah.
I don't know very much about this field.
Yeah.
That convolutional neural net,
Yeah.
basic one. Does it have any spatial structure at all? Like,
It has no spatial, so basically convolutional, so they have weight sharing, that is true. But basically it's a work, I have it here. The way that the network, well, where is this picture? Sorry. So basically these are feed-forward networks, and they basically, each subsequent layer pulls from a local region in the prior layer. Then there's a ReLU, a rectification, and some pooling and normalization. So it's a very standard neural network. So basically what we added to this is actually that, and again, because there's no organization within the layer and it's feed forward, where the units are has no effect on performance, let's say in a categorization task. So what we're going to add is a spatial layout and made it important for the network. Okay, I'm glad that you asked these questions, because if you don't understand the network, it's going to be really not interesting to figure out what's happening later on. Okay. So let's see how the network works. So basically, after we train the network, we fix everything in the network, and we test it on a new set of images to see if it replicates the functional and structural properties of the ventral stream. Since in V1, the dimensions are simple, we feed the networks with these kinds of images that we generated that have oriented lines in different spatial frequency and in different colors. And we want to see first if it generates a functional property of V1 neurons, for example, orientation tuning. So these are examples of different model units in the V1-like layer of our network. And you can see the x-axis is the orientation, the y-axis is a response, and you can see that different neurons are different model units are tuned to different orientations, and also they have varying tuning. Some of the neuron units don't have tuning, but a lot of them look like this. The units also develop tuning to particular spatial frequency. And again, you can see that different model units in the V1 layer also develop different spatial frequency characteristics. So then we can also evaluate how these model units are organized on the simulated cortical sheet. And this is an example of the maps that emerges in our simulated cortical sheet. And basically how this map is generated is that for each model unit, we find its orientation tuning and color coded. So the reds are the horizontals and the greens are the verticals. And you can see that there's a nice orientation tuning. And for the rest of the talk, I'm going to focus on kind of zoomed in regions within the V1 so you can better see the structure of the topography. So here's what I'm going to do. I'm going to show you data from the macaque V1. We need this high spatial resolution, so this is why the V1 data is in macaque. And this is a orientation map in V1. This is a spatial frequency map, and this is kind of color patches in V1 in the macaque data. And this is the organization that emerges in one of the iterations, one of the seeds of our network. And you can see that we have orientation tunings, clustering by spatial frequency, and clustering by color selectivity. Of course, we'd like to quantify this. And one of the metrics that we use is evaluate how smooth are these maps that emerge in our network. So these are the graphs here on smoothness. And we compare this to the actual measurements in V1 shown here in the horizontal lines. And we train many seeds of our network. And you can see that for all of these maps, the TDAN smoothness is on par with that of V1. You can ask what happens when you train the TDAN with just a task, this contrastive task, but without any spatial weight. So we take the spatial weight here is 0. And you can see that the model units do develop the properties, let's say, of orientation tuning or of color preference or spatial preference, but they're not organized in a sensible way across cortex. And again, if you look at the smoothness of these maps that don't have a spatial constraints, they're much lower than what emerges in the brain. So this suggests that both the spatial weight and the spatial loss and the task are important to generate both the spatial and functional properties of V1. You might ask, how much does the architecture on its own really generate these features? So here is an unoptimized map, network, meaning that it's before training, just to see maybe just the architecture on its own will generate these features. And again, you can see that it doesn't really generate the functional features, and basically it has 0 smoothness. It's basically just really, really random. Another thing we tested are this idea of self-organized maps. So self-organized maps are different than deep neural networks, is that you first determine the features of the units, and then what the algorithm tried to do is given a set of features, how to organize them optimally on cortex to minimize the wiring links. And basically, we use 2 kinds of SOMs. One is the SOM that's handcrafted. Basically, you select the orientation tuning and spatial frequency tuning and color tuning of the models. And this is another SOM that we let the network generate the features. And after we've generated the features, we freeze the network and organize this. And you can see that the problem with self-organized maps, whether it's the one that's coming from the DNN or the handcrafted, is that these maps are way smoother than the maps in the brain. And again, if you evaluate the smoothness of these maps, it overshoots the smoothness of the brain. So not only they're not end-to-end systems, but they don't really capture the functional and spatial organization in V1. So how does this model do in human VTC? And again, as I told you before, an organizing property of VTC in terms of its maps and selectivity is selectivity and information about categories. So here we're going to compare it to human data. This is part of the NSD data set. It has high resolution fMRI data in less than 1 millimeter to 14 140 images. These are gray level images that are controlled for low level properties and have about 10 different categories. And among them, there will be pictures of faces, bodies and body parts and limbs, characters and numbers, objects like cars, guitars, things that you can manipulate, and places, including indoor places like corridors and houses. And then we're going to evaluate the function and spatial arrangement of our TDAN by feeding the TDAN the same images that subjects saw in the fMRI experiment. So one of the things that people like to do when they look at distributed representations, they like to look at the maps and see how correlated different pairs of maps are for different categories. This is called the representational similarity matrix. So if I can point your attention here, this is a representational similarity measured from the human brain in 8 subjects from the NSD. And each cell here shows you how correlated a pattern of response, let's say, to a face is compared to a different category, let's say a body. So you can see that faces and bodies are positively correlated, and inanimate objects like houses and cars are also positively correlated, showing some kind of structure in the representation in the human brain. And you can also see that representation of animate categories is anti-correlated from representations of inanimate categories. So we have this really nice or kind of representational structure of multiple category representations in the human brain. You can do the same exercise with the TDAN, so you can look at the vector of responses on the model units in the VTC-like layers and look at how similar the patterns of responses are to the same images in the TDAN. And you can see that there are a lot of similarities. For example, in the TDAN also, we see these correlated responses between faces and bodies. And we see also these correlated responses between places and objects. And we see this anti-correlated responses between animate and inanimate categories. And we can quantify this by measuring how similar are these representational similarity matrix between the model and the brain. And also throughout this talk, when I'm going to benchmark the performance, I'm going to benchmark it to the human-to-human performance. So the NSD data has 8 subjects, and we could see how good one person's brain is a model of another person's brain, because presumably our network cannot do better than using one brain as a model of another brain. So if you compare the human-to-human similarity among RSMs is about 0.7, and you can see that our TDAN replicates a functional similarity pretty well. If you take the task-only model, so this is a model, again, trained on the same and self-supervised task, but with no spatial weight, it's actually interesting that it actually replicates a representational similarity matrix in VTC quite well, and it generates this category structure in the network. Yeah.
Is it a hard-coded thing because it's similarity? The faces,
We zeroed it out because this is like not interesting looking at an image to itself. So sorry, this is a little bit misleading, but here, sometimes we do cross correlation and do split half analysis, but this is actually direct analysis. So the diagonal here, just ignore it, because an image to itself will have maximal correlation. Yeah. Good. I really like it that you asked me questions. So I just want to point you out that even without a spatial weight, we can replicate the representational structure in VTC. But if you have an unoptimized network that's not trained, really it has no category structure. So you really need this task training to generate this category structure. Okay, so what about the spatial structure? Oh, okay, before this, if we look at we also did 2 control networks. One would be the DNN SOM. Again, this is the DNN, and afterwards we try to arrange the neurons and the VTC-like layers. And you can see that this actually doesn't capture the representational similarity as well. And this is another algorithm that was generated by Nick Blouch and Marlene Berman in CMU. And it's called an iterative network. And again, it doesn't replicate the representational structure in VTC. So now let's look at the functional structure. So this is our TDAN, and you can see how the structure looks in the VTC-like layer. And here we're focusing on these handful of categories that we know that they have clustered representation in the brain. And you can see that we get clusters for faces, shown here in red. We get clusters for places, shown here in green. We have cluster for character and a cluster for limbs. So we get clustering of the categories that we expect. And again, we can measure the smoothness of the map compared to the human brain. And this level of smoothness, again, is on par with the brain. If we look at the task-only model, again, it actually generates a representational similarity matrix that's very similar to the brain. However, you can see that even though it does develop model units with the preferred categories, and again, for the representational similarity matrix, how they're organized on the cortical sheet does not really change their correlational metrics. It has no spatial structure. And in fact, this is a task-only model, and this is the unoptimized model. The smoothness is much lower than that's measured in the human brain. And again, the SOM suffer from the problem that they're too smooth, except that the ITN is doing actually pretty well on the spatial structure, but is not doing as well in the functional structure. Yeah.
Do you have any intuition for what the different clusters of faces means?
So I have intuition because we've looked at it a lot with the neuroscience research, and the field has looked at it all. There are two ways to think about it. One way to think about it is that if you have a hierarchical deep neural network, you might have selectivity emerge in multiple regions, and these regions might be tied to perception. And in fact, if we do some kind of a simulated microstimulation in our network, because we have a topography, we can actually do this. You can see that these patches might emerge in more than one layer, and they're kind of linked to one another. If you do stimulation in the real brain, activating these patches does perturb face perception specifically. So we're really sure that they're tied to the perception. It's less clear that they're doing really different cognitive processes with faces. Some people think that maybe more lower regions do more parts and more higher regions do holistic perception. But what I've seen in my research is that there are a lot of metric differences. For example, some patches are more foveals than the others, have bigger receptive fields, but it's not like so simple to put it in like these cognitive buckets that this does one thing and then another does. So from my point of view, they're part of this face network that's involved in processing faces. Yeah. So, okay. So this is a really interesting question. So what determines the number of patches and how much this is about weight sharing? When we started about the project, we really tried to train it without weight sharing. However, when we started this project 6 years ago, our computers were not strong enough to actually do this. But I think today with more powerful GPUs and machines, we could actually do this without weight sharing. In our model specifically, what would affect the size and the number of patches is the alpha, the weight of the spatial loss function. So if you kind of increase the spatial weight, you get more clusters, but then it really breaks the functional property. If you have no spatial weight, then it looks like this and there's no structure whatsoever. So basically, we have, I'm going to show you in the second part of the talk, but basically there's a sweet range of alpha that gives you both a function and structure. So it's kind of interesting that the alpha affects both of them at the same time, because basically, the way we understand it is that the spatial weight affects the representations that are emerging. So you can't really think about spatial organization and functional organization separately. Basically, the sweet spot affects both of these features of the network, basically. And I'll show you that like at the end, the alpha suite. Okay, so what factors, so now that we have a network that works, and this is kind of addressing some of the questions that you have raised, basically we can replicate the V1 structure, let's say orientation, and the category structure in VTC. We can now dig deeper and figure out how much the features of the network matter. For example, how much does the task matter, or the spatial loss function, or alpha matter for the performance of the network? So what I'm going to show you is basically a TDAN and some kind of control networks, and I'm going to evaluate the smoothness of orientation preference map in V1 and category selectivity, for example, face selectivity map in VTC. So one of the things that we were wondering is that how much does the training task really matter? So we've done a self-supervised task because it's more biologically plausible, but a lot of other people think that the role of the ventral stream is to do object categorization. So maybe if we put the categorization in the network with a spatial weight, we can do even better. So this is a categorization task that we did. And you can see that if we look at the V1 like layer, it also produces orientation maps in V1, and the orientation maps are slightly lower, but pretty smooth compared to the ones measured in the macaque V1. However, what was really surprising to us that even though we use the same spatial weight as a self-supervised task, and again, the network does develop these functional representation as well as the selectivity to these categories, such as faces and places and bodies, it actually does not generate any kind of consistent structure in the VTC-like layer. And actually, the smoothness is much lower than the VTC. So this suggests that the training tasks matter for the emergence of the spatial properties of this network. The other thing that we tried is a different spatial loss function. One of our loss function is based on a correlational distance. Basically, it's relative to the size of the network. Other people from the DiCarlo lab, Hio Lee, before us tried a Euclidean distance, which is basically an absolute distance metric. And these are the maps that emerge. And you can see two things that are different about using an absolute distance than a correlation distance. First of all, the tuning of the model units are very different. And you can see that the orientation map that emerges in V1 doesn't look as good as or as similar to the brain. And in addition to that, basically, again, the network does develop these units that are tuned to specific categories, but again, there's no spatial structure in the VTC-like layer. And again, we can quantify this, and we can see that the absolute distance underperforms basically in generating maps and tuning in both the V1-like layer and VTC. So this suggests that both the type of the task that we are training and the spatial loss function is important for recovering the functional and spatial topography of the ventral stream. So now that I've talked to you about the ventral stream, I want to go back to this question about why there are multiple processing streams in the first place. So let me just give you an overview of streams. This is an average brain, and it's shown on the lateral surface. And I want to kind of show you the three streams. So we were just talking about the ventral stream that goes from V1 to the ventral aspect of the temporal lobe. And it's involved in visual recognition, as we talked about. There's another stream that goes from V1 to the parietal lobe. It's called the dorsal stream. And this is a stream that is thought to be involved in processing where objects are in the world. And more recently, in the last decade, we've identified a third stream. It's called the lateral stream. And it goes from V1 to the lateral and superior aspects of the temporal lobe. And it's thought to be involved in processing the dynamics of the visual stimuli or information related to the dynamics, such as actions and social interactions. And what I'm going to do when I'm going to model this network, I'm going to flatten it out. So people can flatten it out with software. And basically, you make a cut on the medial side of the brain and kind of unfold it. So in the pictures that I'm going to show you, this is actually a flat map of the brain. And it allows me to show all the 3 streams in one place. And the dorsal stream is going to be on the top of the map. And the ventral stream is going to be on the bottom. And I'm going to concentrate on these high-level visual areas that's the endpoints of each stream. And my task is to try to explain why there are multiple streams in the first place. So as I told you in the beginning, a predominant theory suggests that the visual system has evolved to be optimized for different behaviors that can be done in parallel. And this is the reason that we have parallel streams. So what we want to do is to test this theory computationally. And the way we're going to do it is that we're going to train 3 different networks. Each one is optimized to produce the behavior that's associated for each stream. And we use this, and the reason that we use 3 different networks is because there are different state-of-the-art networks that are good at doing different tasks. So we use one network to train in a supervised way the network to detect where the objects are in the images. We use another network that is using a data set called kinetics. It has 1,000 videos, 10-second videos of people doing random actions, and it's trained to categorize the actions that is happening in that video. And we turned a third neural network. It's kind of like the network I showed you before, a resonant network that is good at object categorization. So what we're going to do is we're going to take the units from the last convolutional layer of the network, because it tends to be the best corresponding to these higher order areas of the brain, and try to understand where these units are going to be mapped into high-level visual cortex. And the working hypothesis is that units that have been tuned to give you information about object detection might land in the dorsal stream, and units that are tuned maybe on object categorization would best match voxels in the ventral stream. So that would be the way that we are going to test as a behavioral hypothesis. So this is one hypothesis we tested. And the alternative hypothesis is this idea of spatial constraints. So basically, you asked me in the beginning about connections. If the brain were fully connected, the length of the connections would be immense. It would be miles long. So there's no way that we can fit all the wires of the brain if the brain were fully connected into the space of our skulls. So basically, maybe one of the constraint of the biological system is that it needs to minimize wiring lengths. So since we have a model that seems to help us minimize wiring lengths, maybe we could use the same kind of model to explain why streams have emerged in the first place. So we're going to again use this TDAN approach, like I'm going to show you, like I've showed you before, and we're going to look at the last conv layer because it's the most match functionally to high-level visual cortex. And what we're going to do here is we're going to label the units on the simulated cortical sheet according to their location on the sheet. And you can see that I'm using 2 cues. One is kind of coloring by polar angle, like where they are in polar angle, and also dot intensity. So if it's closer to the center, it's more intense. And if it's more eccentric on the simulated cortical sheet, it's less intense. And basically what I'm going to do, I'm going to map again these units in the simulated cortical sheet and see how they are mapped back into the brain, and see if kind of clustered regions in the simulated cortical sheet are going to land in clustered regions in the cortical sheet. So these are the 2 hypotheses that I'm going to test, the behavioral hypothesis and the spatial constraint hypothesis. But one of these things that you could actually see is that in order to test these hypotheses, we have to have a one-to-one mapping between a model unit and a brain voxel, because we want to assign each unit in the model to a single voxel in the brain. Now, this seems like a good idea, but it turns out that we had to develop an algorithm on how to do this, because most ways that people, neuroscientists, compare model units to the brain is using some sort of linear regression or representational similarity that takes a combined outputs of several model units trying to map it to the brain. But if we have this purpose of actually trying to figure out why stream emerge, we have to develop this one-to-one mapping between a model unit and a brain voxel.
Texture is really aware of stream, aware kind of cluster. Is there an object? What do we tell us that corresponds to where the kind of where function?
So the reason that we use it is that basically the output of the network is kind of like a little square, and the image is telling you in this portion of the visual image, this is where the object is. So it actually kind of puts a bounding box on the object. You'll see later on that you can extract other dimensions. You could actually extract metric dimensions out of it, like the position or size of the objects. So it does generate multiple things, but the task that it is to actually identify, put a bounding box around objects in the visual image. So we can talk about it in the end in the Q&A, because there are multiple models that we could have implemented, and we just took the one that seemed to us the most similar conceptually to what the brain might be doing. 10 minutes, okay. I'll try to move faster. So the things that in order to test, to match the brain to the model, we use a really nice data set collected by Kendrick Kay and Emily Allen. It's called the Natural Scene data set. And this is a data set that they conduct, I told you before, they collected high resolution fMRI data for 10,000 images in each subject. So now that we have a lot of data, we can actually start doing real comparisons between networks in the brain. So this is a measurement in the brain. You can see these are the voxels that span all our 3 streams. And basically each voxel, we have a response for 10,000 images in each subject brain. And what we can do is that we could feed our network, whatever network we want, exactly the same images that the subjects has seen. So now we have units by image matrix. And basically what we want to find is the best match between each unit here and each brain voxel. So each unit is only allowed to map to 1 brain voxel. And we want to find some kind of optimal mapping. So overall, the functional correspondence would be best between the brain and the model. And basically, we're trying to minimize this cost matrix. And we want to find this global optimum, and where on average, all the units are assigned to their best functionally corresponding algorithm. And there is this algorithm, it's called the Hungarian algorithm, it's called the Kuhn-Munkres algorithm. And basically, this is the algorithm that we use to find the one-to-one matching between a model unit and the brain. So now that we have a one-to-one match, each unit in the last layer of each model is assigned to only 1 voxel in the brain. We can see, for example, what structure emerges and how does it match. So for example, do all the object detection units map to the dorsal stream, or maybe they're mapped to all 3 streams. So this is basically the mapping between a model and one of our subjects' brain. Again, these are the flat maps. So the dorsal stream is here, the lateral stream, and the ventral stream is here. And you can see that it's a little bit disappointing. It's disappointing for several reasons. Is that, first of all, you can see that in each streams, you have model units from all 3 networks. And second of all, they don't really seem clustered at all. It looks like a kind of salt and pepper. So this was surprising. This is a mapping from our TDAN. And again, this is basically the color coding on the simulated cortical sheet. And basically, we're trying to see how units from the simulated cortical sheet match up to the brain. And you can see that there's a lot more clustering by stream. You can see a lot of red voxels here and a lot of green voxels here. So basically, units that are clustered on the simulated cortical sheet end up being clustered on the brain. So of course, we can quantify this. And one of the things that we quantified is how many units are assigned to the corresponding stream. So this is a different networks that we train on different tasks. And you can see that consistent with the example subjects that I showed you, basically performance is not greater than chance. It's basically only about 1/3 of the units are assigned to the hypothesized stream after train on a particular task. So there's no stream advantage here. In contrast, if you look at our TDAN, you can see that performance is much higher, it's greater than chance, and it's similar to the noise ceiling. And again, the noise ceiling is a brain-to-brain performance. So basically, how many voxels of one subject dorsal stream map to another subject's dorsal stream. And you can see that especially for the ventral stream, we're getting very close to the noise ceiling. But maybe the task optimized model actually provide better functional correspondence. So I showed you the mapping, but how about its generalization to new images? So basically we test the functional correspondence on the left out set of 1,000 images that were seen by all subjects and were not used in the matching. And again, we can see how well the detection, for example, might explain voxels in each stream. And maybe the working hypothesis for the behavioral model is that maybe we have a uniform distribution of voxels of units across stream, but maybe the ones that are aligned actually to the dorsal stream will have a better functional correspondence than the ones that are aligned to other streams. And maybe the TDAN, even though it did generate a nice spatial structure, it would be really poor at explaining the function of the streams because it wasn't really trained on any task associated with the stream because we were still using the self-supervised task that I showed you before. So this is a theory and this is a data. And you can see a couple of things that are really striking about the data. First of all, you can see that the functional correspondence is really much higher for the TDAN than the task trained models. And you can see again that especially for the ventral stream, we're getting really close to the noise ceiling. And this is interesting because this whole network was trained in a self-supervised way in a single task. And even though it captures the different functional properties of voxels in the different streams. Second of all, if you look at the task-based models, it's really, you see no segregation of task by stream. And in fact, across all streams, the categorization task is doing better than the other task, which was, we found really surprising. And again, you can see that it's doing really poorly compared to the TDAN. So we're kind of curious to see what makes a TDAN particularly good. And this is where I'm going to show you the results of the alpha sweep. So we did two kinds of manipulations here to see what features of the TDAN kind of lead to the emergence of stream. One feature is that we varied the spatial weighting, how much spatial weight we put into the system. And the other is that we compared the self-supervised task that I showed you to the categorization task, because the categorization task, even when we have no spatial weight, seem to do better than the other tasks. And on the left, I'm going to show you the spatial correspondence, and on the right, I'm going to show you the functional correspondence, and I'm going to show you that for each of the streams. This is why we have 3 plots here. So first of all, you can see that there's a sweet spot of alpha, and this is the intermediate weights that actually generates the best correspondence, both functionally and spatially, meaning that it governs both properties. And second of all, is that across spatial weights, you can see that the self-supervised task that is more biologically plausible actually outperforms the categorization task that has been used really heavily in the field. So last, you might wonder, well, but there's still different functional properties across stream. Does the TDAN capture this? So one of the things that we can do is does the network on transfer performance mainly, how does it do in other tasks that it wasn't trained for? And the way you do this is you freeze all the network weights and you just train a classifier as the output of the network on a task that you're interested in decoding. And again, we chose 2 tasks that are associated with the ventral and dorsal stream. We had one task that has to determine the position of the object in the image in kind of metric units of the image, and a categorization task telling us how well this performs. And this is on the TDAN that was trained with a self-supervised task, so there's no explicit training of the network. And what we really find is that there's this really nice double dissociation. So we trained these tasks either on the units that were assigned to the dorsal stream or units that were assigned to the ventral stream. And you can see that units that were assigned to the dorsal stream do better at the position task than the ventral stream assigned units. And conversely, units that were assigned to the ventral stream do better at the categorization task than the units assigned to the dorsal stream. So we see this double dissociation, suggesting that even though the network is trained with a single task, clusters of the units develop different functional properties that might lead to the emergence of different behaviors that people have measured, for example, neurally and behaviorally. So to answer my question, why is the visual system functionally and spatially organized? Our results seems to suggest that as the brain is kind of optimizing learning generally useful representations in a self-supervised way, together with a spatial constraint that minimizes wiring length. And using the same kind of principle, you can see emergence of function and structure in many spatial scales, from the orientation columns in V1 that are about maybe 200 microns big, to clustered regions that are about centimeter on the side, to the whole emergence of the stream architecture across lobes. And one kind of interesting insight from our work that we didn't anticipate, that we're implementing really a local constraint. The spatial constraint is local, but it can percolate up the system and generate this large scale organization like whole processing streams. And it also suggests that it's important to model not only the function, but also the topography of brain representations if you want to glean insights into the underlying mechanisms. And of course, this is just a beginning because we've only examined certain features of the function and the spatial topography. And you might want to code other aspects of neural function like spatial temporal processing or other aspects of topography, such as cortical magnification, to provide a more comprehensive understanding. And one of the things that my lab particularly is interested in is in modeling spatial temporal dynamics and incorporating them into deep neural networks. For example, we have a recent paper that we use fMRI to measure the spatial and temporal receptive fields of neurons in the visual system. And you can see that there's really a nice hierarchy of both temporal window increasing as well as spatial window increasing across the visual processing hierarchy across three streams. And the other reason that we're really interested in this is that when we did our comprehensive sweeps, for example, across task and across alpha spatial weights, we can see that we're doing worse in the lateral stream compared to the other streams. And one of the reasons that we suspect that this might be the case is that NSD is a static data set, but the lateral stream really responds quite vigorously to the dynamic stimuli and video. So we think that if we had a dynamic stimulus set, that we have really big brain data and we have a spatial temporal TDAN, maybe we could start closing these kinds of gaps. So with these ideas, I'd like to thank you for your attention, and I'd be happy to take questions. Yes.
Thanks for the talk. It's really interesting. One difference between the last methods that you're comparing for like 3 networks, so not only is also the hypothesis that you basically have a very late splitting in the TDAN case, right? Meaning that like all the components of the lateral, dorsal, ventral streams are shared until the very end.
Yeah.
And so my understanding of the visual system is that you know, we think that happens in V1 and V2, but then after that, things start getting separated out.
Yeah.
I was wondering if you could just sort of comment on that.
So this is true. So if you look at this picture here, you can see that this is what you call early visual cortex. This large rectangle is V1, V2, and V3, and that is shared across 3 streams. And you can see that these are smaller rectangles here are these intermediate visual areas that are assigned to each stream. So there are intermediate visual areas that are assigned to each stream. So we didn't train these more elaborate networks, and it's a really good point that you make that might improve our model. Another thing that we think that is going to be really important, and maybe replicating other aspects of the topography, is or maybe here, I'll go back to this, is actually implementing the magnification factor. So if we look here, V1, I've looked a lot of these maps, so I know you have to trust me that I'm telling you the truth. The center of V1 is the fovea, and the periphery of V1 is here. And there's much more real estate in early visual cortex to the center than the periphery of the visual field. If you look at the ventral stream, there's also an organization by eccentricity, where phase selective regions are tied to foveal representations and place selective regions are tied to peripheral representations. So one of the things that I highlighted is the success and maybe less so of our failures. So if we go back to this picture, for example, of the ventral stream, you can see that the place area is kind of in between the two face patches. And I can tell you, this is not the way it's organized in the brain, where the face patch is on one side on the lateral VTC, and the place patch is on the medial VTC. So and because we haven't implemented this cortical magnification, one of the hypotheses that we have that implementing, again, another features of the biology, for example, you wanted me to implement these intermediate regions. I want to implement also the eccentricity, cortical magnification. Maybe that would actually generate the patches in the right order on the simulated cortical sheet. So we're some aspects of the map we're producing, and other we're not. Yes.
I really love your idea that wiring length is a constraint on brain processing. Can you say more about where that constraint comes from and how it works?
So how it works, I don't know. I've generated a model to implement this. So this is kind of how we've implemented it. So maybe this one. The idea is that if the brain were interconnected, like everything would have been connected, it's basically the connection lengths would be miles long. So you can't really fit it into the brain. The other thing is that it's also metabolically expensive to have all these connections. So a biological system has real physical constraints. So for example, we all know that OpenAI has, I don't know how many computers that can reproduce large language, or Google might have tons of computers that can do really good at face recognition. But these are really, how do you call it, energy expensive, right? And we somehow survive on between 2,000 to 3 calories a day, right? So we have to build, when you think about biological systems, you have to build efficient systems. It is also the case that different animals have different kind of evolutionary niches. So what the humans use a visual system might be different than a mouse. So again, there are different kind of evolutionary pressures on different animals. So the idea is that there is a real physical constraint in a biological system that some of our computer systems don't really have the same kind of pressure. And there's a lot of evidence that the connections in the brains are pretty efficient. They're probably not as efficient as the feed-forward network that I've implemented. For example, there are also recurrent connections within layers that I haven't implemented. There are also these long-range connections that I've measured myself, and they're not implemented. So we have a very impoverished network. So what we can do in our model is actually minimize the within layer connections, because it's a convolutional neural networks. But what we can do is think about how it would be implemented in our network. And this is why we have this kind of putting neurons together, units together that have similar responses close together, is a way that we've implemented it computationally, because we don't really have these local connections in the network. The other papers that we implemented the algorithm is the ITN by Nick Blauch. They really tried to do within layer wiring. The problem with their model is that they can only do it in one layer. So they don't really have, how do you call it, a single principle that they can implement in all steps of the network and actually show that wiring length minimization actually generates a whole ventral string. So basically, different people are approaching it in a slightly different way. And what I like about our model, of course I'm biased, is that we have a unified model. It has the same principle and it's trained end to end. So we don't do any kind of like futzing and optimization at the end. And I think that's kind of the way that I feel more comfortable doing my science. Yeah.
This is a more methodological question, I guess, but so you built this model where you introduce the spatial loss,
Mhm.
and then you have this metric for smoothness that you use to sort of like, I guess, validate the model. Do you have any other metrics that are, I guess, were surprising or emergent? Because it doesn't seem surprising, I guess, if you like introduce a loss to make things close together, but they're close together.
So we've looked at multiple metrics of the networks, and if you look up our, it's in press right now, but you can look at the by archive version. You could see that each figure has like many panels, and each panel implements a different metric. So for the purposes of the talk, I'm just showing you like the basic metrics that we've implemented, but we looked at a lot of functional properties and a lot of spatial properties. So for example, for the face patches, for the patches in the VTC, we look not only on the smoothness of the map, but also the distances between the patches. We have an automatic algorithm that generates if you find a patch or don't find a patch. So you could see, like you can measure clusteringness. You can look at the spatial relationship between the patches. So we've looked at multiple metrics, but I chose for purpose of simplicity and not to be overwhelming, kind of like the most obvious metric. And I'm really curious to hear your thoughts. The other thing that for the VTC data, we have a lot of data in my lab, but for the V1 data, it's what people have published, and kind of we have to work with what people have actually measured. But if you have any ideas about other metrics that we could use, I'm really interested in hearing about this, because some of them we had to invent as we went. Yeah.
What are the way to, like, right now, there's a loss function to get the model to learn spatial structure. But is there a way to trap this or build it into the model in the first place and then see how it performs?
Okay, so you want us to hardwire the spatial constraint.
Sorry.
Okay, so as Roxanne said in the beginning, a lot of my work actually does a lot of measurements in the human brain. And one of the things that's really intriguing for me is how the brain develops. So we always look at the adult, like this is like the gold state, but turns out none of us are born as adults. And we kind of like worked our way into that over like maybe 18 or 20 years. So that has caused me to look at children's brains and also look at babies' brains. And the more I'm looking into it, it really changed my way of thinking about it, because when we think about it computationally, we take a network and we just tune the weights. But when I look at the brain, it actually grows. So connections form, also connections prune. There's actually physical changes to the brain anatomy. So when I've seen this empirical data, it really causes me hesitation to actually hard code anything. I would like, there's some basic architecture. So like the number of brain areas probably is there. The general topography and connections are there, but the details, I think I would like the network to learn. So this is kind of why the empirical data suggests that I shouldn't really hard code that. Maybe one last question. You had a question over here. Somebody here had a question. Yeah.
So I was also wondering about, so the difference between the set of different downstream connections as well. For example, the OST. So you commented all on sort of how that fits into your.
So I'm really interested in measuring these connections, and we do a lot of diffusion works to actually measure the connections of these different patches in these high-level cortices to downstream and also to early visual areas. And again, right now, all I showed you is a proof of concept. And again, I've used a very simple network. It's a feed-forward network. I know that's not biologically accurate, and basically is the goal where we're going with this research is add additional, more biological physical properties. So one would be let's say, the eccentricity bias. The other is to actually incorporate these long-range connections. We're probably going to start with long-range connection within the visual system, but it would be also interesting to implement downstream connections to other cortices that are involved in other behaviors. My colleague Dan Yammons really think, and this tied to the baby question, that one of the things that make us human is actually our embodied existence. And the way we learned about the world is not just from seeing things, it was touching it, interacting with it, hearing it. And that's kind of where these downstream connections to other cortices really play a role in actually shaping the representations in the visual cortex as well.
Thank you. We should probably stop there. Let's thank. I believe she's going to be around for the day, so if you have more questions, she'd be happy to talk to you.
Yes.
For the next